I will start to introduce Raji. So I'm very happy to have today here with us Raji Balasubramanian. I met her really long time ago. She's doing a very advanced statistical tool for solving important and critical problem, complex disease, and using graphical network modeling for genomic and not only genomic, biological data in general. So yeah, the floor is all yours. Thank you.
Roberta, thank you so much. I just wanted to say thank you so much for inviting me and for hosting me here today. Had the privilege of meeting several wonderful PhD students and new faculty, and also loved seeing some familiar faces in the audience as well. So I'm so excited to be here. Thank you again. This is work with Dr. Kate Schuta, who is now at Harvard School of Public Health. And this was work she did while she was a PhD student at UMass. And my talk is on this method that we developed called the SpiderLearner, which is the goal of which is to estimate in sort of a robust fashion a network given a data matrix of continuous data, such as in a gene expression study. So I am hoping to sort of go through sort of these chunks during this talk. Just provide a little bit, very sort of high-level overview of networks in medicine. So for those of you who may not be familiar with how networks are used, just to provide a little bit of context. And then I'll review sort of the foundational sort of material on Gaussian graphical models, also very briefly, but just really point out the key results that we use in developing our estimation of our network algorithm. And then I'll spend some time talking about how we set up our model framework and estimation, provide some brief results from simulation, and then present results from applying our method to some data from an ovarian cancer database. So, so how many of you have sort of worked with or at least learned about in classes about biological networks in various applications? Okay, many. So I'm kind of preaching to the choir, so I'll keep this quick. So obviously, networks are widely found in medicine and biology. So we can think of social networks that comes to mind immediately sort of after experiencing COVID-19 together. So modeling human connections, pointing to shared environmental or behavioral factors. Or we can think about networks of diseases. So the goal there would be, are there like really distinct disorders, but share some common underlying, aspects, either genetic or otherwise, environmental factors. And then cellular networks are the kind of specific kind of cellular networks are the kind that we have a lot of experience with in our group. And they can be networks involving gene expression data, protein, either abundance or protein-protein interactions. Those are quite different data types, or metabolomics data, which are like gene expression data, but they're measuring these small molecule metabolite abundances in various samples. So just as a reference, I found this article by Laszlo Barabasi about 20 years ago, just talking about the promise of network medicine. And I think it's a really nice overview, especially if you're new to this field. And before I launch into our particular sort of method and its application, I thought it's also nice to sometimes see an entirely different way in which a network might be used and how you can glean some insight. So this was a study that was published in 2007, also in the New England Journal of Medicine. And they analyzed data from a subset of participants from this large cohort called Framingham-Hertz study. So I don't know if people in EPI, we hear about Framingham-Hertz study a lot, but if you haven't heard it, it's one of the oldest cardiovascular cohorts established in Massachusetts, in the city of Framingham. And the primary goal for establishing that cohort was to study risk factors of cardiovascular disease. And it's still ongoing today. In this 2007 study, they looked at social networks in a subset of 12,067 people, and they assessed their social networks at various time points for which they had data. And they were trying to understand the spread of obesity in social networks. So obesity is a condition that has a strong genetic underpinning, but they saw some remarkably strong and striking associations just from social networks. So this picture here is a network of individuals where each node is a person. They're colored either green or yellow based on whether their BMI is over 30 or under 30, and that's the WHO classification for obesity. And the size of a person's node is just proportional to their BMI. So the bigger circles are individuals with higher BMI. And what they saw is that at every time point, they created these social networks. Discernible clusters of obese persons were present in the network at all time points. And when they modeled it, they saw that a person's chances of becoming obese increased by 57% if they had a friend who became obese in the same given interval. And this is over, this effect is actually eclipse any genetic effect that people have found for obesity. So it was really interesting. And it kind of struck me to bring into this talk, not because we're modeling social networks here, but just to point out that networks are cool. You can come up, you can use these tools to really analyze all different kinds of data that can give you interesting insight. Okay, so stepping back into sort of our work, I'd like to just go over a little bit about Gaussian graphical models and how they apply to the kind of data that we're trying to model in our application. So we're thinking of, just to make this a little bit more concrete, we're thinking of a data matrix which has some number of rows that represent individual features that you're measuring. So those could be genes, individual genes, and some number, and the columns then indicate either individuals or samples in which those genes are measured. And the colors of each cell, ranging from red to blue, indicate whether the expression of that particular gene in that sample is really high if it's red, or really low if it's blue. And the rows, and so this is a standard heat map representation showing that the rows and columns can be clustered. And in many real biological data such as this, you see this kind of pattern where subsets of genes cluster together. And what we mean by that is that they have similar expression patterns across the samples. And so you can see right off the bat that here are a couple of genes that are all highly expressed across our samples, and the opposite is true for these genes. So all of this is to say our data is a matrix of some number of features or genes measured on some number of samples. And these are continuous values because, as the title of my talk suggests, we're using the Gaussian distribution. So that's only appropriate for continuous data. And we're trying to apply networks because we believe that these genes don't act independently of each other, but they're connected to each other. And so we want to use a network to represent their connections. So, so then just showing you in a picture what I just said in words. So what if we take our data matrix and then have a network where the nodes are our genes and the edges between our nodes are undirected. So there isn't an arrow that's going from one to another, but they're just simply connected to each other. And that connection is some measure of a pairwise strength of relationship between the nodes. So what are Gaussian graphical models? So Gaussian graphical models are a special type of undirected graph where we assume that the nodes of our network come from a multivariate normal distribution, and that's the reason why it's called Gaussian type of graphical model. And the edges that connect pairs of nodes represent pairwise conditional dependence. And what do I mean by conditional dependence? So then, again, formulating the Gaussian graphical model, just using some notation, we have our nodes. Let's say we have P nodes in our network. So we have a vector of nodes. These are our random variables of interest. We assume they come from a multivariate normal distribution. There is a mean associated to characterize that distribution and a corresponding P by P matrix, which is the variance covariance matrix. In a Gaussian graphical model, the edges are weighted according to the partial correlations between the pair of genes. So partial correlations are correlations between the gene pair, but conditioning on all of the remaining genes in the network. So in other words, another way to think about that is that a partial correlation adjusts for all of the other genes in the network and then asks what's the measure of the direct relationship between the two genes that we're connecting or not by an edge. So here's a simple example of how a partial correlation might work. So let's say you have a set of 3 genes. And let's say that I'm going to generate some data for gene 1 using a very simple linear function. I'm going to say that the average value of gene 1 is going to be 3, and I'm going to add some random error to it. Okay. So gene 1 is not influenced by others. Gene 2, on the other hand, is 2 times gene 1 plus some random error. And so because of this expression, gene 1 and gene 2 are positively associated. If one goes up, the other one also obviously goes up. And then gene 3 is inversely associated with gene 1. And so through this simple linear equation. And so obviously, if this goes up, this goes down, and vice versa. However, in this setup, we have not said that gene 2 and gene 3 are connected to each other in any direct way, right? So the equations are all either gene 1 by itself or gene 2 and gene 3, depending on gene 1. So if I generate data from this framework, and then I just look at the marginal correlations of the data, I see what I expect to see between gene 1 and gene 2 and gene 1 and gene 3. Those correlations are positive and negative. But then I also see a strong inverse correlation between the genes 2 and 3, even though neither of them was actually directly influencing the other through our setup, our data generating mechanism. But if I take a partial correlation instead of just the regular old marginal correlation, then immediately this strong negative edge that I observed in the correlation world goes away in the partial correlation world because there really is no direct relationship between X2 and X3 once I've accounted for all of the effect that flows through X1. So that's what we mean by a partial correlation. So when we are creating a network, we want those edges to be weighted according to the measure of the direct relationship between the pair of genes once you've accounted for all of the associations that operate through all the remaining nodes of the network. So just to contrast, you may have seen correlation networks. They look similar, but the edges have a very different interpretation. When you just look at them, correlation networks for biological data tend to be like a hairball. They're highly densely connected. And that's because the marginal correlation is the sum of both direct and indirect relationships, as we saw in that little toy example. But once you condition away all the indirect relationships, a partial correlation network ends up being much more of a sparse network characterized by fewer edges because those represent only direct relationships. Okay, so the goal is we want to be able to estimate. We want to be able to estimate a network like this given some observed data. And many of the methods, the graphical lasso and all of its variants, exploit a very special property when you make the multivariate normal assumption. So again, let's say you have p nodes in your network, and we assume that the p-dimensional random vector follows a multivariate normal with a variance-covariance matrix given by sigma. The inverse of this variance-covariance matrix also has special properties. It has a name associated with it. It's called a precision matrix. And so if sigma is a p by p matrix, so is this one. And we refer to that by the letter theta. And the special property is that if you can estimate your elements of the precision matrix, then you can directly estimate all the pairwise partial correlations through this expression here. So this is only true for under the assumption of a multivariate normal distribution. And so the foundational work and all of its variants exploited this relationship that given an estimate of the precision matrix, you can directly estimate the partial correlations, and from there, you can estimate the network. So they transformed the estimation of the network to be a problem of estimation of the precision matrix. And so that's what we work with going forward in the SpiderLearner. So we ultimately want this, but we will get there by estimating this matrix up here. Okay, so in 2008, Hasty, Friedman, and Tisch proposed this method called the graphical lasso. It's closely connected to the lasso if you studied that in class or used it in elsewhere. And they proposed this approach by posing a likelihood function based on the multivariate normal distribution assumption on our p-dimensional random vector. And so if we assume that the data are centered, so we can assume that the mean is 0, the log likelihood for our observed data is of this form, shown in equation 6. So I want to point out here that we have two unknown parameters. The first one is theta, which is our precision matrix. It's the same theta up here, or the inverse of the variance covariance matrix. And that's really what we're after. We want to estimate that given a set of data. And then our likelihood function obviously has to be a function of our observed data. And that comes in through this matrix S. This is the sample covariance matrix. And so our likelihood function is a function of the observed data through the S here. And the other parameter that is important is this parameter lambda. And that's analogous to the lambda that shows up in the ordinary lasso. So if you've seen the lasso, you know that it's a penalized regression model or a regularized regression model. And the penalty term lambda is a non-negative constant that governs the sparsity of the estimated precision matrix. So larger the lambda, the more sparse the entries of your precision matrix. So once you have the log likelihood, and if you have an approach to estimate the optimal lambda, then you can maximize the log likelihood and obtain an estimate of the precision matrix, and from which we can calculate the partial correlations, and that yields a network, right? So it sounds easy, but it can be tricky. And one aspect of what can be tricky is the algorithm that goes with maximizing this log likelihood. The other is how do you come up with an optimal lambda in a data-driven fashion? So because of these, from the original timeline of when the graphical lasso was published, there's been a series of innovations and improvements on that original algorithm. And these have had to do with papers where they have proposed a variation of the algorithm for providing computational improvements in estimating our precision matrix from that same log likelihood function, and many others that use different scoring criterion to identify what's the optimal value of lambda. So while we have one model and one observed data log likelihood, we end up with a variety of ways of estimating the unknown quantity of interest. And so, which brings us to our work, which is, okay, if you have a bunch of data, which one am I going to pick? So consistent with all of the work that we have, at least this is not all of it, this is not exhaustive by any means, but these are many of the key papers. There is also software packages associated with it. So when we started doing this, we were like, this is an interesting approach. We have several data sets, just let's try this approach on our data and see what we find. And here's an example of some real results from a example data set, where each of these are different ways of estimating the same matrix theta, the precision matrix theta, and the corresponding network from the same data set. So you can see that right off the bat, they look wildly different from each other, right? So then we said, okay, maybe we can do some kind of simulation study, think of all the different sort of types of networks that might exist in real data and see if, which one, is there something that consistently outperforms the other? Can we provide any practical guidance to saying you might want to pick this if your data looks like this and so on. But really, in reality, we could find nothing that seemed like an obvious winner in all situations. All we were left with is that these estimates for the same data set, estimating the same underlying precision matrix, can look really different. So our goal is to come up with an approach that can combine these various estimates. So if you have a library of different methods that you want to try to estimate the underlying precision matrix, and you have all of those m different estimates, how can I combine them to form an ensemble in a data-driven way in which the performance of the ensemble is at least as good as the best method in your toolbox? So this actually is a lot like a prediction approach. Oops. Okay, this is a lot like, so we derived our motivation from learning about the superlearner for prediction algorithms. So the superlearner says, let's say you want to classify, I don't know, images with cats from images from dogs, and you have all this data, and you have so many machine learning algorithms. You have random forests, XGBoost, you have neural networks, what have you, right? And can I come up with an ensemble of all of my favorite algorithms such that the ensemble does at least as good a job as the best performing classifier in my library? So we approached it in a similar fashion. So we want to know if you have m different software packages that each does the optimization and estimation of theta in a slightly different way. And so you have m different theta matrices estimated. Then can we learn from the data a weighting scheme, a convex combination of these, so these weights, alpha 1, alpha 2, and alpha m, can we learn them from the data such that the ensemble, which is a linear combination of these estimates, performs as well as the best one of these methods? So we call this our SpiderLearner estimator. So it is some weight alpha 1 times the first estimate of theta plus all the way up to alpha m times the mth estimate of theta. And so our problem now becomes, if you have a library, we can estimate these thetas from these m different approaches in our toolbox or library. So the problem that we are solving here is, how do you estimate these alphas that tells us how to combine these estimators together to come up with a linear combination or a convex combination? Yeah.
Can I ask you one question?
Yeah.
The previous slide.
Yeah.
So this a superlearner that was introduced by this van der Laan.
Yeah.
Was done in the graphical network or for general
prediction algorithms? Yeah. Yes, exactly. Exactly. So if you had lasso and you had random forests and XGBoost and you build all those classifiers, how do you combine them into a convex combination to come up with a superlearner?
Generally, theta 1, theta 2 is a vector, or could be a matrix?
It would be, so if you're predicting case versus control, for example, in n subjects, it would be a vector of predictions, or yeah, right, exactly.
Variance or
right. It's a prediction rule. So in the superlearner, it would be a prediction rule. In our situation, it's a matrix, an estimated matrix. Yeah. Yeah.
Okay.
Yeah.
Thank you.
Okay. So again, we propose a estimator of this form where everything but the alphas are estimated already from the data. So the next step is, how do you learn the alphas? So we propose this cross-validation approach. So let's say you have your full data here, and let's just for the sake of argument that we are doing a 5-fold cross-validation process. So then you would partition your data into 5 non-overlapping folds or pieces. And let's say this is the first iteration of your cross-validation, and you hold out the 1/5 or the red piece of your data, and then you use the remaining 4/5 of your data as your training data set. So from this 4/5, let's say we have in our library these 4 different methods that we want to combine together into our superlearner in some way. So we take our 4/5 of our data, and then we apply each of our methods one at a time. And so with each of these methods, we get an estimate of this underlying thetas, which is theta 1, theta 1 hat, theta 2 hat, theta 3, and theta 4 hat, right? So we can loop through our cross-validation through its five iterations, and we arrive at 20 different estimates, because in each of our 5 folds, we have 4 estimates of theta based on the 4 methods in our library. So they're arranged in a matrix of this form, where you have this column are the estimates of theta from this first cross-validation step, which were trained on just this group of data, and this was left out as an independent test set. Yeah? Okay. So then to figure out how to estimate our alpha, our superlearner is of the form, in the first iteration, sum alpha 1, which is yet unknown, times the estimate of theta from our first method in the first cross-validation iteration, plus alpha 2, plus alpha 3, plus alpha 4. So to come up with the best guess for alpha, we write out the log likelihood of the test data set, so the data set that is shown here in red, given this, where this theta matrix, the true parameter, is estimated by our superlearner, expressed as this weighted linear combination. So in this fold, we can write our log observed data log likelihood based on the multivariate normal distribution. So it's a function of our observed data that is in the red square, and a function of our, a function of these 4 different estimates of theta, also generated by the training data here. And then a function of the only unknown parameters in this function, in this loss function, which is alpha 1, alpha 2, alpha 3, and alpha 4. Everything else is known once you condition on the data in that iteration. So we then repeat this process across all the folds and average the results to obtain our objective function. And then we're left with a function of the observed data, but where the unknown parameters are just the weights that we want to learn. And we can maximize our log likelihood using our out-of-the-box solver and nonlinear optimization approach, and then estimate the alphas. So for a given data set, our SpiderLearner algorithm is we go back to the full data set. So now we have the full gray vertical bar. We re-estimate the 4 different thetas from the 4 different methods in the library, and then weight them by the weights that we just learned through the cross-validation process. So that is our final ensemble estimator of the SpiderLearner. So we did extensive simulations trying to understand whether this works, and is it worth doing? Obviously, it's quite computational because of all of the k-fold cross-validation, and because you have, depending on whether your library is really big, you can be fitting many different approaches to estimate theta in each of your cross-validation. So it is pretty computationally heavy. So we did this simulation where we ranged our sample size n from 10,000 to 60. We considered a range of number of nodes of our network, and we considered a range of network topologies from random to small world scale free and hub and spoke models. So if you're not familiar with what these mean, I'll show you a picture in the next slide. I can show you that these networks just look really different in their structure. And the reason people studied them is certain types of biological data often look like very different from what you would call a random network, and but have characteristics of these other networks that are shown here. We also considered graphs where the true graph is really dense versus others that are less dense. So here are the graph topologies. The top 4 are where only 6% of all possible edges exist in the graph. And the bottom 4 are where 20% of all possible edges in the graph. So already with 20%, you can see it's starting to look like a hairball. It's starting to look like a dense network. On the left-hand side is a random network. So this means that every pair of edge has the same probability to exist in the network. So if you were creating a random network, you would plonk down all the nodes, and then you would say, and if I want 10% edges in my network, then I would assign an edge to every node pair with 10% probability. So that's just a random edge, and that's what one iteration of something like that looks like this. Contrast this to something like this, which is a hub and spoke model. So here, these networks are characterized by few nodes that are connecting a lot of the other nodes in the network, whereas most of the nodes in the network have very few connections. So there are these, so in social networks, we all know people like that. There's always the person who knows everybody. And most people have, a few friends, right? So I think, so you can think of a biological network that's an analog to that here, such as a hub and spoke model. So all of this is to say we did a simulation assuming the true network looks like a variety of different things that you may see in different applications. And we asked, does our SpiderLearner algorithm work equally well in all of these situations? All right, so we compared in our simulation the ensemble estimator to all of these individual approaches. Again, for example, these approaches are all based off of GLasso. We call them different because they actually end up estimating different looking networks on the same data. And that's because they're choosing their penalty parameter lambda in a slightly different way. So, and then we also have the hub graphical lasso, which is a variant of the graphical lasso that is very particularly tailored to networks that actually look like this. So we have these various individual approaches that we can sort of compare the SpiderLearner estimator to. So I won't go into this in much detail, but we looked at several different metrics of thinking about what is good performance versus weak performance. And so here's one such example. I'm sorry, this is a little fuzzy. But in the setting where you had, in the setting where you had P equals 1,275, that means you have a network of 1,275 nodes. You have a network of 50 nodes, and you're estimating 1,275 pairwise connections, or 50 choose 2. And let's say you have 10,000 people in your data set, then our simulation results shown here shows that for 2 different types of graph structures, this is a random graph, and this is an hub and spoke graph. And we're looking at a measure called the relative Frobenius norm, which is a measure of how close our estimate is to the truth. So in that context, a relative Frobenius norm that is really low is a good thing because it's really close to the truth. And something that's up here is worse. And then we have all of our methods lined up along the x-axis. Circled in black is the SpiderLearner. And right next to it is just the simple average of all of the m different estimates. So we want to know, is it even worth doing all of this, jumping through these hoops to estimate the alpha, or would just a simple equal weight to all of these work just as well? So that's why we threw that in there. And then we have all of our other individual methods, their relative Frobenius norms lined up right next to it. And so you can see that in both these situations, for example, here, the hub and spoke, when the true graph is of hub and spoke, it's no surprise that the hub graphical lasso does really well, right? But so does the SpiderLearner. So you would say, why would I need the SpiderLearner if I know that this is doing such a good job? Why do I need one more method that does the same thing? The problem is, in a real data application, you can never judge which one is working well because you don't know the truth. So what we want is an approach that will work at least as well as all the tools in a library, regardless of what the truth is. And so that's why having this approach is a useful additional method in your repertoire. So the takeaway is that both in low and high dimensional cases and in the range of network topologies, it is able to perform at least as well, if not better than the best candidate method. It definitely, we conclusively decided that it's better than the simple mean across all the simulations. And it provides us a practical way to optimize this decision-making process on which method to pick. The last piece is an application to ovarian cancer. So what I'll do, so we actually, in our paper, we took a data set, we estimated a network, and then we estimated sort of communities within the network. So communities are connected subsets of nodes that are highly enriched for connections to each other. And then we asked, are they enriched for certain biological processes that make sense in cancer? And the answer was yes, we could find that. So it was sort of a descriptive sort of exercise in applying our method to data and seeing if we find some interesting biology. The other that I thought was we both thought was really interesting is what if we take our network, use it to inform a prediction score, and then validate that score in completely independent data. And actually, we were able to do this in several independent data sets. So if you're looking for some high-dimensional data to play with, this is a really nice package that has 16 data sets from 16 studies, all on ovarian cancer cases. So let's see. So I am going to skip ahead to this. So what we did in the training phase is we picked one of our 16 data sets. So this is a data set that came out of the study by Yoshihara et al. It has 260 late-stage ovarian cancer patients. And the outcome here is survival. So they where they had surgery following chemotherapy, and then they had information on how long each patient survived beyond that. And what we want to do is use these data, and we wanted to use a subset of the genes, 126 genes available in about 20,000 genes, to create a survival score that we then want to take to independent data sets and see how well it predicts survival in completely independent cases. So, so we chose a subset of 126 genes here because of the 20,000 genes, there was previous literature that showed that these 126 genes were associated with survival. And so we chose to form our network using that subset to have sort of more manageable set of data to work with. And then I'll explain in a little bit more detail. We created our network using these 126 genes, and then created a survival score that was informed by our network. And then associated with a survival score, we came up with a threshold to classify individuals into someone who's high risk versus low risk. And then in each of our 15 sort of independent validations, we know how to calculate the score based on the training data. So we calculated using the same rule and the same set of genes. We calculated the score in each of these 15 data sets. And then we also know the threshold of the score that corresponds to low versus high risk. And so we grouped individuals in our independent data sets into high versus low risk using the threshold that we obtained in the training phase. So now we have in each of our data sets 2 groups of people, high risk and low risk people. And so we can compare their survival using an analog of a 2-sample t-test or the log rank test. I don't know how many of you have taken survival analysis, but it's an approach similar to, is an approach for comparing survival between 2 groups. So in each of our validation data sets, we end up with a p-value comparing the people we designate as high risk versus low risk, based on the gene survival score that we estimated in the training phase. So So how did we create the survival score? So we take our Yoshihara data set, we calculate our network using, SpiderLearner, and then we take our network and identify communities within our network, and within each community, we figure out which gene is operating like a hub, which gene has the most connections to everything else in that community. So then we take our, instead of all the 126, we focus on just the hub nodes, and then create a survival score by fitting a model to our training data set. So now I have estimates of beta 1, beta 2, beta k, and from fitting our model. And then we take it one step further, and then we ask, what is the value of our score that effectively separates those with low survival from those with high survival? So from this training phase, we get estimates. We know which ones are the hub genes. That's important. And we know how to weight them. We know the numbers that correspond to beta 1, beta 2, and so on. And we also know the value of S that will let us classify a future person into low versus high risk. So when we take that into our validation data set and then compare our low risk versus high risk individuals, this is the result that we got. So let's focus on the bottom bar graph. The x-axis is p-value. So it's the p-value for comparing those who are called low risk to those who are called high risk. And we had 15 data sets that we validated our score in. So we have 15 p-values comparing our low risk and the high risk individuals. And our score in this last row was estimated using SpiderLearner to estimate the network. So it chose 7 hub genes to make the score. And you can see that of all of these bars, this one is the closest to 0. So these p-values were all highly significant in these 50 independent data sets, saying that the score that we developed that was informed by the SpiderLearner estimate was doing a pretty good job in separating those with low survival to those with longer survival. And in contrast, we tried the same approach, but we replaced the network estimate by each of the individual methods in our toolbox. And you can see that first of all, many of the methods picked a lot of hub genes. Like, for example, the top one picked 42 hub genes. So you have a pretty complicated score. But then when you use that score to classify low versus high risk, you found p-values that were pretty large, which means that that score is not doing a great job in separating those who have low survival from those who have longer survival. So we thought this was a neat illustration of how you take a network and you translate it to predict something that everybody cares about and see how well that prediction works. Here's just a picture of the survival curves for some of those 15 data sets. To summarize, our ensemble approach, we believe, flexibly accommodates a user-defined library of estimation approaches. It works at least as well as the individual estimation approaches. And in an application, it was really useful in identifying a sparse gene set that was pretty powerful in predicting survival in completely independent data sets from that database. So this is, we have a software package that Kate has in her GitHub repository. So if you're interested in trying it out, please do check it out. And Kate has also put a lot of thought into developing a very well thought through manual and user guide. So definitely, hopefully enough to get started. And if you have any questions and if you have interest in learning more or applying this, we'd of course very much welcome your acknowledgements. The lead author of this paper is Dr. Kate Schuta. Our collaborators are Laura and Denise at Berkeley and Northwestern, and we gratefully acknowledge all the participants in the ovarian cancer studies. Thank you.
working from my interest on applying this.
Yeah.
Do you have a sense of how robust the method is if there is missingness in the data?
That's a great question. I believe, so our method starts from when you have those M estimates, right? So a lot of those, the GLasso package, for example, will not allow you to hand in a matrix that has any missingness in it. So you would have to sort of impute the data before you, sort of send it down this pipeline. That's a great question.
Say, pick up the estimator for the graph.
Yeah.
You said that it's just the estimator, but in this study, you just pick up that GLasso family best estimator. Would that be like, is that good to pick up? Like, I'm not sure whether using families would work, but is that using more? Because I know that like GLasso is more based on the graphical Gaussian like this assumption, but there is some graph based on other assumption. Is that like including the graph estimator for other assumption will improve this work?
That's a good question. So we also do use the Gaussian assumption, because in our cross-validation approach, our loss function is a multivariate normal log likelihood loss function. So I think in that context, if you had in your library an estimation approach that is, let's say, makes other parametric assumptions, I'm not sure is consistent with sort of this framework because of the type of loss function. But if you take one step above that, and if you talked about just the idea of an ensemble and picking a loss function that is appropriate and going through the cross-validation process, I think then absolutely you can expand your library.
Great talk. I have several questions. So the first question is, we know the gene expression data, the raw expression is just still the count value. So the normality assumption might not satisfy here. So how the model is sensitive to like deviate the normality assumption?
Yeah.
How does it?
Yeah, so I don't believe we looked at deviations from the normality assumption, because we sort of assume that our library will have, as tools, the graphical lasso, which makes that assumption. So, however, I think when you have count data, when you have or zero inflated data, they don't work well, like in practice. So obviously, but if you had somewhat skewed distributions, you can try sort of transformations and the like to sort of make it work. And I think it's fairly robust. Obviously, I think but if your data type is different, then I think you run into trouble.
Next question is, do you have any assumptions on the weight? Because like sparsity assumption on the weight to make sure that the weight is not overweighting for some specific method that is more close to your leave one out samples in your cross-validation.
That's a great, no, we don't actually. So that's, some of the limitations or maybe avenues for future research is we don't we entirely choose, everything is equally likely as suggested by and then, weighted up or down based on what the data says. We're not using any prior information in choosing, in sort of estimating the alphas. The other thing is that we weight an entire matrix by a single number. So, that's also a pretty strong assumption. So
yeah, I'm thinking about because for some data, maybe the some network inference method probably just capture part of the network.
Correct.
But, just reading everything together.
Yep.
It might introduce some bias. So
yep.
Like what you said, maybe by consider more about module-based weights for the network.
Exactly. Yes. Exactly. So like blocks of the matrix, and yeah, that makes a lot of sense.
You made the survival risk score, you said you made the subset of 126 genes.
Yeah.
How did you pick the genes, and does it matter which genes to pick if you were going to repeat it with a different data set?
Yeah, so that was simply for illustration. So we picked the 126 because of the prior literature showing that that is a signature of survival in ovarian cancer. So, so it gave us sort of a convenient way of like narrowing our focus and not having to deal with like a really large network. But if I was doing this purely from an application point of view, I wouldn't sort of drastically drill down from 20,000 down to 126 without like strong rationale for doing so. Yeah.
How extensible do you think could you add proteomics data in concert and things like that?
I think so. As long as the multivariate normal assumption is a reasonable one, I think for sure. Yeah, there's no reason not to.
Yeah, just one last question.
Yeah.
Have you seen this method in a Bayesian analysis? Like, and this is related even to Ingmar question.
Yeah, yeah.
Never, like even for like prediction or linear models or something like that.
Yeah, I haven't. In the network literature, you know, at the time we were working on it, there was definitely nothing that existed even to, as you know, like the network estimation approaches is like more new than prediction classifications. So certainly there isn't a Bayesian analog that I know of.
Okay, let's thank again our speaker.
Thank you. Thank you so much.